Hello, I'm Gabe Ibanez, and this is my sustainable development goal. In particular, these pertain to number 15 and 11. Number 15 is to protect and restore and promote sustainable and terrestrial ecosystems, substantially manage forests, combat desertification, and halt and reverse land degradation and halt biodiversity. So mine uses plants, and it actually has to deal with some biodiversity, artificial actually. But number 11 is making cities and settlements inclusive, safe, resilient, and sustainable. So basically these are gonna light up, fluoresce the area around, similar to that of lamps within the city and how they light up the streets. Except for these will produce a more warmer, less bright, but still will produce enough light to where you can be seen. The roads can be seen. Okay. So, what is this prototype lab? Well, basically, I'm gonna try and enhance plants with a bioluminescence characteristic. There's several forms of this, and they include, there's two main types. The first one is photoluminescence, which contains fluorescence and phosphorescence. In fluorescence, that is basically energy absorbed and the emission of light when that energy falls from the reaction. That's commonly found in a lot of animals. Some important things about this is it only fluoresces when the energy source is actually present. So for instance, if the sun is not present, then the light will not occur. And some of this is also regulated by the GFP protein, which is also known as the green fluorescence protein. And I'll get more into that a little bit later. But some important people that actually created this thing and actually won the 2008 Nobel Peace Prize was Martin Shalfie, Osamu Shamuro, and Roger Cian. And they won this by presenting the GFP. And now today there are several forms of that in RFPG, YFPG, or YFP, all fluorescent proteins. So the chance of actually doing this has a semi-high possibility. There's some modifications that one may have to do. The next one is phosphorescence, and this is where organisms store energy in electrons and give off photons over a longer period of time, meaning that instead of it being right as the energy source dies, light will not be produced, but once the energy source dies, there's going to be a little bit remainder of energy left over by the fall, causing the luminescent feature to occur. So the second main type of luminescence is chemiluminescence. The two subtypes are man-made chemiluminescence and bioluminescence. So man-made chemiluminescence is where basically electrons get excited, they fall, and reactions ending up in light. So there's some important factors are the oxygen reduction process, which helps emit light by the energy fall within the reaction. So next there's bioluminescence as the second subcategory, and it's a type of chemiluminescence, and this is actually found in like jellyfish, certain bacteria and fungi around, and possibly can be transferred into plants such as these. Some important factors of this is the light is produced via a chemical reaction within the organism, and luciferin, it's a chemical compound that light is light emitting within the organisms, and this is very important, and I will get to this very soon. The next one is incandescence, and this is basically light being emitted due to heat structures. So if you heat up a room or you heat up the temperature, surrounding cells will actually activate and cause this bioluminescence feature to occur. So what I'm doing currently, because the other processes of luminescence are a bit costly and I don't have the kinds of material and money to get those, it's called a black light method. And basically this means there's a fluorescent material but doesn't shine unless there's a UV light. In this case, a black light is being used. So some items that cause this fluorescence within black light are tonic water, which I'll be using here, chlorophyll, which actually turns red under the black light, which, as you see, it's green because that's what our eyes are set. But niacin and riboflavin, which are also found in vitamins, actually happen to glow. So the possibility of this actually happening in terms of putting it in the real world of black light will have to have some serious modifications, more so with bioluminescence, but this is just a little preview of what can happen. So the important terms that I told y'all I would get to is the GFP and luciferin. GFP stands for the green fluorescence protein. It has been regulated to show other colors like red, blue, green, yellow, so many more. And now luciferin is a chemical compound found in bioluminescent organisms. And when oxidized with the help of luciferase, light is emitted in the chemical reaction. And so how it functions, there's three main things that I'd like to see that could be able to be a possibility in the future, is phosphorescence, bioluminescence, and just regular fluorescence. Now, for phosphorescent, chemicals would have to be added to the plant diet and monitored over a course of months and days with different variables to see how the phosphorescent material reacts with the plant. And the second possibility within that category is one can possibly inject that material into the organism's surrounding cells so that there's a possibility of the substance being replicated via meiosis and mitosis. Now, the second version is bioluminescence, and this, as said before, is found in many animals, and it's caused by the luciferin protein. Now, there's a possibility that one can alter the genome of a plant structure by inserting and DNA splicing this gene and adding it in, which will then be activated when using the luciferase gene via oxidation. So there's now the second version of that is, if the main thing that I'm trying to hit at with this one is if the luciferin gene can be inserted into these plants, there's a theoretical possibility that the organisms, when given the oxidative conditions, light will be emitted. The terms in how bright that light will be exhibited have not been determined yet, because don't have the materials, but plan on doing that. Now the last one is fluorescence, which kind of deals with bioluminescent organisms, is the GFP. And that has been done recently, but it's actually been put into Rhabditosis thalina, a tobacco plant used for many of the science chemical additions and regulations, and GFP was actually added into there. There's some photos online if you just so happen to look it up. It does fluoresce a bright green like chlorophyll as seen in here. Okay, so the prototype that I'm doing, it's plants with light and there's bacteria, but it would be really cool to see some photosynthetic organisms containing this light, which could actually possibly replace street lights where we're using electricity, and it causes a lot of light pollution in the society, which messes with human circadian rhythms, animal circadian rhythms, and plant circadian rhythms, which actually take a major toll on how nutrient-rich the plants will produce and how organisms will interact with others. So most of the nutrients that will be required by these plants are water, of course, moisture, nitrogen, which actually makes the leaves green on a plant, potassium, which helps the plants fight off diseases and bacterial invasions, phosphorus, which is for making the flower blooms strong and very more thick and robust roots, and proper temperature and air quality, of course. Okay, so the materials I'll be using today in this lab consist of silver daisies, which, if you look back here in this cardboard container, are silver daisies, and right here are all impatiens, several different plants of them. We'll be using tonic water. I used two types before in making this lab, but now I'm going to use the Refresh, which also works. I've used them in and out replaceably, have the same effect. And also I'll be using this 75-watt black light, as seen already, plugged in. We have tried it many times. It's worked. We'll be using this tablespoon measuring cup. It's for ingredients like eggs and stuff, just at-home cooking materials. Some dyes, specifically blue, yellow, and red. Okay, so what we're gonna do, because I have already put a lot of water into a lot of chemicals into these already, the tonic water, in fact, I'm actually gonna do a lighter dosage. In fact, I'm gonna do 4 tablespoons to be exact, and so each of these, I tried to make them color-coded the best I could. The control group, which I thought may be a little cool to add some more variables to this, is this pot back here where it'll just have normal tonic water in it. The rest will have each cell have tonic water, but there will also be a certain dye in it. I just wanted to do this to test the possibilities of seeing if the dye would actually change the fluorescence patterns, which you'll find very interesting here in a couple minutes. Okay, so first thing you do is you take your tonic water, you unscrew it, make sure it doesn't explode on you because it is carbonated, has some carbonation in there. You're basically just going to pour the water in there till it reaches about 4 tablespoons. I used 16 just to see if I could get more absorption by the plants, and it worked, but you want to make sure you don't overdo this. So I'm going to fill each cup first. So this is the blue dyed cup, as I have already done this before, and I'm honored to have some calculated results in a presentation I have made to go along with this. Oops, forgetting steps. So now you're just going to continue filling up. Each cup will be filled up first, and then I will put the control group in, then dye each of the cups. So it's about 4 tablespoons. It is a somewhat lengthy process, but it's relatively fast and results come out soon. So I have the 3 variable cups going on right now. I'm just gonna let those finish out. I'm going to pour the control group's substance in right now. And basically you just pour it in. Now, tonic water is a mixture of sugar, water, and CO2, and a few other chemical compounds in there, which actually tonic water was used to treat malaria diseases back in the history of the world. So now that we've done that, I decided to go with about 6 drops of each dye. Not too much to where it's overused. 6. So the yellow one can also be seen as orange sometimes in the cups. Just gonna mix them up real quick. Make sure that the dye spreads evenly throughout the cup. Okay, so if you want to come look at these real quick, you got the red, your yellow, but it kind of looks like orange, and you got your blue. These cups are actually used from an AP Bio lab that I had previously done and taken home some of the plants just for fun, see what I could do, which got me started on this. So I'm actually gonna now turn off the lights and actually show y'all what happens when the black light reaches these cups, and I think you'll be surprised, and I'll put these in and show you how the plants are affected. The effects actually don't occur right immediately, but these are just from earlier waterings that you can still see the luminescence products within some of the stems, not so much on the leaves, but that'll have to be modified later to possibly work. So I'm going to turn the lights off, and so the black light is on, and you may not, the camera quality won't pick this up, but the plants are actually going to glow red because of the chlorophyll within them. And if you look over here at the cups, which were once red, yellow, blue, are all now showing the same fluorescent feature. Okay, so the next part. This is a pretty easy step I like to consider. So the red goes with the pink. You basically just pour it into the plant. You want to make sure that the plant itself gets the water in the container. It will fizz up with contact with the soil, but it will go down eventually. So I'm not doing the silver daisies currently because they have been super saturated with my previous watering earlier today because they are in a smaller container. But if you come up close, you can actually see some of the fluorescence. There we go. Some of the, as seen, it comes up as a light pink kind of shine luster within the stems right there. If you come down here to the other group, you can see actually right up in this area there's some shiny fluorescent thing. It's not coming out as well. I wish it would come out better. There's some more things. If you'll come over here to the red, you can mostly see it back here in this back root area. Yeah, so some there. You may have to zoom out. So you get a fluorescent, iridescent kind of look on the plant stems as seen right here. And that's basically the plant absorbing the tonic water, but you want to make sure you don't over fill up the tonic water to make sure that the plants are receiving an ample amount of nutrients but aren't oversaturated. This process hopefully can be used in the future and maybe modified with bioluminescence and chemioluminescence, and unfortunately I did not have those kind of materials with me because they are pretty expensive and I don't have the money, unfortunately. But I'd like to further research this because it's very fun and interesting to watch as plants show off little features under black light. The main goal of this is actually to replace the lights in our city, because you drive down your neighborhood street every night and you'll see lampposts about every 100, 200 feet. And some of them are capped to where it only shines on certain areas, but most of them it shines all around. And this is not to replace street lights, but mainly the plant lamps just that go across the streets, electricity. So basically the emitting of the light in our city creates light pollution, and if you go actually up into some mountain ranges or in fields where it's farther away from light, you can actually see glare, somewhat of a light haze that is shone in the sky basically at night, and it blocks scientists from doing research on solar planets and galaxies. It makes it harder to think. As well, earlier stated, it messes with circadian rhythms and electrical use. We use a lot of electricity, whether it's just powering the lights that are on right now or the black light that I just used, charging your phones. It actually creates a reduced amount of that, and if we were able to insert this experiment on these plants and it was to work, we can introduce this into trees that would fluoresce at night so that you'd have a somewhat warmer color but not as emitting and bright as these, to where if you look at them you'll probably have retina damage, same as looking at the sun, which would help the society in reducing a lot of pollution as well as electricity use, and make the environment a better place. Thank you for watching. Hope you enjoyed.